Uh, Welcome, everybody, to our weekly HEALEY platform update. Today, we have a special guest, scientist from Prilenia and the co-leads of the Regimen D for the platform trial. I'm Merit Sukovich, chair of neurology at Mass General Hospital and the principal investigator of the HEALEY ALS platform trial, and also here with Sabrina Paganoni, the co-principal investigator. I want to thank our patient advisory group and all of you who come to our weekly meetings for your suggestion that we invite each of the scientists and leaders of the companies that are working with us on the platform trial to share the excitement behind their product and the target, and to answer your questions about their development plans for ALS and in particular their drugs. So our first group is from Prilenia, and they're going to tell you about Pridopidine. But before we go there, just for those of you who might be new to coming to this webinar, I just want to give a few high-level overviews of the platform trial. And apologies for those of you who come every week and you've heard this from us before. But we really wanted to reimagine how treatments are developed for ALS and really speed up the process and borrow an approach that's already been highly successful in different diseases of cancer. And that's called a platform trial. And it really speeds up the time to getting to effective treatments in half. It increases by 2/3 the chance for people to be on active treatment, and it lowers the cost of drug development by 1/3. And traditionally, one drug was tested at a time. In the platform trial, we test multiple drugs at a time. Each person is assigned to one, but because of the efficiency of the platform trial, we can pool the data from people who are on placebo, and we can quickly add new regimens, and we can also quickly remove regimens. And it's a way to be adaptive and responsible and flexible, so we can accelerate ALS therapy development. And we're really excited to launch this first platform trial in ALS in July of 2020. With a lot of people's help, a lot of people's support, and also in the middle of a pandemic. And that really speaks to all of you and the ALS patient community about how we work together to not let anything get in the way of finding treatments for you. Next slide, please. So just to visually show what this looks like, when someone comes into the platform trial, they're randomized to one of the available drugs that is in the platform in that time. And right now we have 4 different drugs, and you're going to hear about one of them today, Pridopidine. And then after that, so let's say you're assigned to regimen A, you'll know that you're assigned to regimen A. And then there's another randomization where 75% of people will get the active drug and 25% placebo. And each group has 160 people. And at the end of the 24 weeks of the study, we take everybody that was on the active drug. So again, let's say for regimen A, 120 people on the zilucoplan, and we compare them to everybody who was in the placebo in each of the arms, which is 40 in each. And so at the end, we're really sharing that placebo group, and that's where we're getting the economy of time and savings and increasing the percent of people getting active drug. And each of our partners has agreed to provide what we call open label extension to any of the participants in the regimen. So let's say you finish your 24 weeks on Pridopidine in that arm, then you have a choice to go in open label extension. In open label extension, there's no placebo, and this will go on for a couple months until we have the results of the double-blind trial. And then if it's positive, there'll be a decision about whether that continues. If it's not working, then we would stop the open label extension. There would be other options to discuss with the participants. So that's the overall design. We don't get the results of the study, and this has come up in some of our other webinars. We don't get the results of the study until the last person is enrolled in that arm, let's say in the Pridopidine, the last person in Pridopidine finishes 24 weeks, then we can unblind and look at the analysis. Throughout the study, though, there's a safety committee that's always looking at safety. And they meet about every 3 months, and they do know who's on drug and who's on placebo, and they can weigh in if there's ever any safety questions. Next slide, please. Each of our regimens is led by two physician scientists. And so the Pridopidine one is led by Dr. Jeremy Scheffner, chair at Barrow Neurological Institute in Phoenix, and also by Bjorn Oscarsson, who's a physician scientist, neurologist, ALS specialist at Mayo Clinic. And they're here too to answer your questions after we hear from Dr. Hayden from Prilenia. And with that, I'll pass the Zoom baton to Dr. Michael Hayden from Prilenia, who's a great neurologist and physician scientist, to tell you about Pridopidine.
Thank you, Merit. I'm delighted to have this opportunity to talk to everybody today and to share with you some of the exciting news on Prilenia. I wonder if you could just move the slide forward, please. It's not showing on my screen. So we want to share with you about Pridopidine for ALS. We're very excited to be part of the platform trial. It's really been very inspiring to learn and see what has been done for cancer and then see what can be done here for ALS. And we've learned a lot, and Merit and Sabrina, the leaders of this trial and the catalysts for this transformation and innovative trial, have really been the drivers, and it's a great privilege to work with them. But I think the trial offers a hope and opportunity for transformation of the future. Transformation of bringing new and innovative drugs in a quicker way for the urgent unmet need, which is to patients with ALS. Next slide. So when we look at some of the questions, and I know these are questions that we've been asked before as to this particular drug, and I'm going to try and answer each of them and give you some perspective on why we're excited about assessing Pridopidine in ALS. The first question, of course, is what is Pridopidine? And we are going to go through each of these questions and try and provide some answers to give you information so you know why we believe in this drug, why we think it's a good opportunity to assess in its potential mechanism and opportunity for results in ALS. Next slide. So when we look at what is Pridopidine, Pridopidine, next slide, is a small molecule, so it's taken as oral therapy, and it's an investigational drug right now in clinical trials in ALS and also Huntington disease. You may say, how could it be for both indications? Because even though the cause for each is different, the pathways that are activated as part of the disease leading to neuronal death are similar. And Pridopidine is administered orally twice a day, once in the morning, once in the evening. And it binds, we now know the target of Pridopidine, it binds what is called the sigma-1 receptor. This is a receptor present throughout the brain, but particularly expressed in the motor neurons of the spinal cord and other regions of the brain that are relevant for other indications. What is useful is there's a lot of experience with Pridopidine, and it's safe and tolerable and actually has a placebo-like tolerability profile. And the dose tested here for ALS is the same that has been tested now in 1,300 patients for about a year. And so it's a long history, and it's similar and has a profile that's very similar to placebo. And in patients with Huntington disease, we saw what were very promising and early but exciting results is that this showed maintenance of function. It showed less progression than seen in patients on placebo based on the mechanism of activation of the sigma-1. We also saw that if you look at patients who've been followed for up to 5 years and been on the drug, that their progression was much slower than that seen on placebo, what would be expected. Furthermore, there is in Huntington disease, there's similar to ALS, there's an accepted scale that can be used to assess function and progression. It's accepted by the regulatory agencies and can be used as a single primary endpoint. And just similarly in ALS, there's a functional scale that is the similar endpoint, the same endpoint in all the trials, looking at the ability to slow progression of disease. Next slide. Now, what is the sigma-1 receptor? And the sigma-1 receptor, next slide, is in fact a protein that's highly expressed in the brain and spinal cord. And I said it's seen particularly in motor neurons, particularly in the spinal cord. And it plays an important role in modifying stress. In fact, when you activate sigma-1 and it's activated either by a ligand or what we call the drug itself, pridopidine, it activates it. This drug, this particular receptor, interacts with numerous targets in the neuron, and it promotes neuronal health, it reduces death of cells, it enhances neuronal health by also improving the response to stress, so it decreases the stress response. It improves the respiration of the cell by improving mitochondrial function, and the mitochondria are the breathing apparatus for the cell. It also is important in improving connectivity. What's been quite remarkable when you look at animal models, you can actually see increasing connectivity in the brain, and increased connectivity, so the neurons have spines that connect with each other, and it enhances this, and it reduces neuroinflammation. So it does a lot of good things, and we've seen similar results. Next slide. So you may say, well, what do we know about the sigma-1 receptor in ALS? And why is it a good target for ALS? So as we said, of course, it's expressed in those regions of the brain that are involved in ALS, particularly the brainstem and spinal cord. And here you can see blue would be low expression and red would be high expression. So there's high expression in the cortex and particularly in the spinal cord. And next slide, and the most exquisite validation comes from human genetics. And one of the questions you ask yourself is, are there patients with ALS who have genetic spelling errors or mutations in this gene, the sigma-1 receptor? And lo and behold, there are families now well described. These are families who have either complete loss of the sigma-1, and interestingly, they get a very early form of ALS, what's called juvenile ALS, even before adulthood. And then those patients who have decreased function as a result of mutation, but it's not zero, it's just decreased, these patients get late onset ALS. So this was exquisite genetic validation from different parts of the world that told us if you have a loss of function of sigma-1, you cause ALS. And then, of course, there's animal models. Next slide. There are animal models that are important. These are ALS mice with a particular genetic mutation. And what you can see is that essentially, if you look at the animals who have both the gene that causes ALS deleted, but they also have a double mutation and they have absence of the sigma-1, their disease is worse. So these animals in the red line, they survive less, and animals that just have the mutation without sigma-1 live longer. They have increased survival. So removing the sigma-1 accelerates the disease. This gives you good validation that sigma-1 represents a very validated target in humans. Next slide. But that's not enough, because now we know that loss of function causes ALS in sigma-1. But of course, here we're looking at most patients with ALS who don't have mutations in sigma-1 and don't have mutations, but have other causes for ALS. What happens when you overactivate and you increase the activity of the sigma-1? And here we see that if you activate sigma-1, you get some neuroprotection. And neuroprotection, and these are all factors that have been shown in many different publications. Firstly, you get clearance of toxic proteins, and in ALS, you do get accumulation of toxic proteins. You increase the energy production, which is decreased in ALS. The spines are improved. You get enhanced neuronal activity and connectivity. Importantly, it leads to secretion of what we call neurotropins or neurotropic factors that promote neuronal health, called BDNF. It regulates iron levels so that they are normalized, and it decreases cellular stress and decreases inflammation. So all of these have been shown, and let me just show you a few examples in pridopidine in ALS mice, where you can see some of these effects. If we could go to the next slide. So a crucial part and junction in ALS is the connection between the neuron and the muscle. And the muscle here is shown in red, and the neuron is shown in green. And you can see on the left, you see a very nice interaction essentially between the muscle and you can see the muscle and the neuron just as one example. But if you look at the top, you see, in fact, there's dissociation between the neuron and muscle, where the arrow is pointing to the muscle. The green is not really present. And what you'd like to see is that when green and red are superimposed, you get yellow, which means that they now are interacting with each other. And what you can see here, using pridopidine, you get nice interaction. The neuromuscular junction is rescued. The connection is rescued, as shown in the yellow slide. And furthermore, when you look further and you see some of the cells and you look at the number of neuromuscular junctions that are now innervated, these are increased with pridopidine compared to animals that have ALS alone. So this, again, these ALS cells show increased innervation, precisely what we're hoping to see in ALS, where we can improve the connection between neurons and muscles and improve the connectivity. Next slide. But furthermore, you really want to look at the function itself of the cells. And if you look at the little squiggles there on the right, what you can see at the bottom, you see a contracting myofiber that can give you very strong response in terms of intensity. And above it, you see an immobile myofiber where the intensity is much less, the intensity of the variation of that those signals that you see here. So healthy cells show, similar to the bottom, nice contractility of the neuromuscular junction and the muscle itself. And that's seen on the right, in the figure, what's called wild type normal animals, 70% of their muscle fibers can contract. Now, in an animal model for ALS, what you see here is you get disruption of this. And now you see less muscle contractility, and that's the middle bar, the purple bar in the middle. And these animals really have much less, down to 50%. And you can see many of these animals have immobile myofibers seen at the top. And then you add pridopidine, and it improves it and restores it back to normal. So these are, of course, they're animal models. They're not, mice are not humans, but they give us strong indications and reason to believe. So I'd say it's mechanism of action, the validation genetically, and then the ability to see how pridopidine would actually improve function in different in different neuromuscular junctions. That particular junction gives one reason to believe, and the compelling reasons to really do this particular study. Next slide. Now, of course, everybody wants to know that these are safe. You know, as physicians, we firstly want to do no harm. We want to make sure the drug is safe, and it has been tested in a lot of people. Next slide. Here you can see it's been tested in 1,300 people for about a year. So that's a long time. There's a lot of history, and most of them were patients with neurodegenerative disease, especially Huntington's. And there've been a lot of studies that have been done, and some of them have run for 5 years. So we've got also knowledge about the long-term impact of in the brain. And in fact, we now know that this is really safe and tolerable with no long-term side effects at all. Now, another question is, how do we know in humans, next slide, that pridopidine actually gets into the brain and spinal cord? How do we know it's going to reach its target? A drug won't have its effect unless it gets to where you want it to get to. And this is a slide that shows, firstly, you determine this by using a marker. And fluspidine, seen above, is a marker for the sigma-1 receptor. So there you can look at without pridopidine, and you get very nice labeling. The yellow and green shows that sigma-1 is present throughout the brain, and you can see this labeled in this particular figure. And then what you do is you now know that if you give fluspidine, it'll go to the sigma-1. Then you give a drug that potentially could block it, and that would be pridopidine. And you ask yourself, how much is the signal decreased? And what you can see here, quite remarkably, that the signal, when you then give fluspidine, is completely blocked. The door is blocked for labeling the sigma-1 receptor because it's already occupied by pridopidine. So pridopidine very quickly gets into the spinal cord and brain exactly where we want it to go. Next slide. Well, another question to ask is whether there's any evidence that pridopidine can slow progression of other diseases. Do we have any reason to believe from other diseases where the drug has been used? And we will show you, this is early data, so it's what we call phase 2, mid-stage drug development, that gives you the basis for doing a phase 3 study, which is now ongoing for Huntington disease. And this is using TFC, which is a measure similar to the functional scale in ALS. And when you go down, you get worse. And what you can see that patients on placebo go down as expected about 1 point per year. That's exactly what you expect based on all the natural history studies. But I would say surprisingly and remarkably, the patients on pridopidine did not deteriorate over 1 year. And that was very exciting data that led to the approval for a phase 3 pivotal trial, both in Europe and in North America, both America and Canada, for that particular indication. Now, next slide. HD has a lot of similarities to ALS. I mean, sadly, both of them don't have very effective therapies to slow down the course of the illness, which is the whole purpose of the platform trial. But also patients themselves talk about what's most burdensome to them. And it's the loss of function, the inability to do activities of daily living, the inability to continue working, the inability to hug your children or your grandchildren, the inability to just perform daily human tasks and be playful in ways that you want to be physically. And so patients have stressed that the burden of loss of function is really what's most important. The scale in Huntington disease measures some of these things, and the ALS-FRS scale in ALS also measures not exactly the same because the disease is different, but also measures important functional activities that we are looking for and seeing if there's any improvement in ALS. The biggest issue for patients with Huntington disease is they don't want to be dependent on others. They want to be able to maintain their own honor, self-respect, and independence for as long as possible. And of course, that is something that's so needed for many neurodegenerative diseases, but particularly for ALS. So when you look at this, we are seeing that next slide, that many research over a significant period of time have shown the beneficial effects, and the effects appear to be significant and long-standing. And that's why we're so excited about doing this study in ALS. Just to go back to the safety again, next slide, just to give you a bit more detail. We know the drug is safe. There's been now extensive studies, as we said, 1,300 patient years. What a patient year means is that one patient, 1,300 patients have been treated for the equivalence of a year. So it's called 1,300 patient years. They've had numerous doses, much higher than we're using in this dose, 45 milligram BID. or a total of 90 milligrams a day, but this has gone as high as 225 milligrams per day. The drug appears to be safe and tolerable, and the dose to be tested in ALS is the minimal dose of 45 milligrams BID, where we've got a lot of data on safety, and the side effect profile is comparable to placebo. Let me just give you some of the data so that you can actually see some of the data that has been looked at in terms of the similar incidence of side effects. Next slide. What you can see is these were some of the side effects that were reported, the most common side effects in placebo. And what you can see there: falls, nausea, nasopharyngitis, diarrhea, headache, and dizziness. And when you look at that, you say, well, you know, we don't really want those to be a side effect of a drug. But when you compare this frequency of this to placebo, none of them are really significantly increased. You know, the falls are more or less the same. Nausea actually higher than placebo. Headache equivalent. Diarrhea essentially the same. So these are really common and not really attributable to the drug. And so the side effect profile, and this is at the dose that we are using. Next slide. So we're excited to participate in this landmark trial. It's a highly selective, it is the most selective sigma-1 receptor activator, because you don't want a drug that has a lot of promiscuous interaction with other targets. You want it selected because we know that that's the target that we need to reach. This is a remarkably selective target. It's validated because genetically loss of function is associated with ALS. It's also neuroprotective in animal models. Gain of function by using activity of the drug has beneficial effects. We know this drug gets to the right place where it can have its function, and we know it's safe and durable. And all of those, this compelling evidence supports the therapeutic potential of pridopidine in ALS. So we're excited to be able to participate in this important trial, and I'll hand over to Merit to continue this important discussion. Thank you. You're muted, Merit.
Sorry about that. I was speaking for a while. Thank you, Michael. We now have about half an hour for Q&A. If people, if you have questions for Michael or for Jeremy or for Bjorn or any of us, please put in the Q&A. I'm going to read them out loud. So the first one is maybe for Michael. If a patient has sporadic ALS and genetically caused retinitis pigmentosa, would you postulate that pridopidine could benefit both diseases?
That's a brilliant question. It's we have separate data in retinal degeneration because through evolution and millions of years, the pathways have been conserved, even though the cause is different. Once you get to a neuronal injury, the pathways of cell death are conserved. And ALS and Huntington disease has activation of pretty much the same pathways, even though the cause is different. And the same goes for retinal degeneration. So we've looked at that, not clinically, but in animal models, and we find that we can get significant reduction in retinal loss and retinal degeneration in various animal models. These are animal models where glaucoma, the pressure is increased and you induce retinal degeneration and you get dramatic loss, a dramatic decrease in retinal degeneration. So this is a whole nother discussion about the use of the drug more broadly. But yes, I wouldn't say that's a contraindication to being in the trial. And there is certainly at this point theoretical potential benefit.
Thank you. Next question, maybe Michael, you could start, and then Jeremy or Bjorn, I would love your thoughts on this too. If the neuron muscle connection is increased or improved with pridopidine, would this reverse or restore muscle atrophy that has already happened? And did you see any of that in your animal trial?
Yes. Well, you know, it's the clear, so the answer is we don't really know. But if cells have already died, it would be very hard to regenerate them. On the other hand, if there's injury and it's not dead, the cells haven't yet become atrophic and died, there is a possible that you can decrease the injury. The one thing that I think and that could be resulting in some improvement. In Huntington disease, for example, there were a proportion of patients who actually had improvement in their function. So they started off at a certain scale and they actually ended up showing improvement. But that was about a quarter of the patients on treatment. Most patients were just maintained, they didn't go down. So I would say we would hope to see a failure of progression, and an upside would be some improvement, but I'm not sure that we could expect that. And we're going to this very open to measure it, but without any presumed evidence for that at this moment.
I don't know if Jeremy, you wanted to add anything, maybe not about the drug, but the general concept.
Sure. I mean, so if you see evidence on pathology that there's disruption of the connection between nerve and muscle, it can mean a lot of things. It can mean that the neuron is just dying, as Michael just said. And in that case, it's the idea that Pridopidine, or really any other drug, is likely to meaningfully impact that is low. It could also mean that the neuron is still alive, but maybe not totally healthy, and that the part of the neuron farthest away from the cell body where all the energy is produced is becoming less stable and impacted in some way. And in that situation, if you can increase the general health of the neuron, you potentially could restore some of those connections. We know that in ALS, many motor neurons who are still alive and will be alive for a long time still are unable to contact as many muscle fibers as a normal motor neuron can. And so there is the potential to increase this ability. And to the extent that that's increased, you could imagine that this would cause an increase in strength. So it will be interesting to see.
Thank you. How close is the FDA approval for Huntington?
The approval, well, of course, we're doing a definitive phase 3 trial. And I'm getting some echo, but and so that trial will be reading out at the end of 2022. So later than the ALS trial, because it's a longer trial, progression is much slower. And if you want to see an impact on progression, you have to go longer. So there's a window between the placebo effect and what you see in a treated group. But the FDA and the EMA have been very supportive. Firstly, they've said, yes, you can go into a pivotal phase 3 trial. They've said significant results would be approvable. But of course, so I would say we're somewhat away. If we were to get results, this would be and probably the end of 2023 approval. So it's still a long way away. It could even be a little longer than that, depending on the length of regulatory assessment. Europe usually takes a little longer. So in the US, end of 23, early 24, Europe in 2024.
I'll just add that in Huntington's disease, sometimes the trials are longer than in ALS. So it takes longer to get readouts.
Yes, maybe just to explain that in Huntington disease, for example, all the measures read out over a minimum of 1 year. There is one other phase 3 trial going on in Huntington disease, sponsored by Roche, which is an antisense trial. And that trial goes for 2 years. So it's even longer, because that means you have to recruit patients, and then the last patient has to be followed for 2 years. So that's at least 3 years to get results. This is shorter because we've already shown within 1 year that we can see results.
Thank you. Bjorn, I wanted to give this question to you with the caveat that it's a really important question, but it's also really a hard question. And that was whether there's any promising research anywhere to develop drugs to stop the progression of ALS, not just to slow the progression.
I mean, all these studies that we're doing in the platform would be able to detect such a market change. These studies are done with a setup where we will be able to see a smaller change. I think that any drug that works on one of the downstream effects of the disease alone probably does not have a high likelihood of stopping or reversing symptoms, but quite possibly a combination of things. And we are now adding, I mean, riluzole, edaravone, and now several different compounds in the platform that are in trials, as well as other studies that have recently completed phase 2 that might be making it to the US market. So maybe with a combination of these, we can really get somewhere very powerful with these types of drugs.
And I wonder if I could add something here. Again, we can learn from cancer, because in cancer, it's very seldom that you use a single drug for treatment. Combinations of therapy have been very effective. And this platform trial is looking at single agents in each arm. But if we were, for example, to show, some benefit for the 1 or 2 arms, and there was a rationale, one could look at combination therapy that may actually be synergistic or further enhance the effect. So if we look at other models for other diseases, combination therapy is not unusual and is often the best, most effective approach.
Yeah, I agree completely with both of you. And in the platform trial, we do allow people to take standard of care, which could be the riluzole and the edaravone. So we're starting to get that combination.
Yeah.
And so this is for you, Michael, which is, did I hear you correctly say that the sigma-1 receptor can be activated by light?
No. So we know that sigma-1 receptor is activated by that was a ligand, which is actually, and the ligand is just the chemical in your body that activates it. Actually, for this, we actually aren't even sure what the natural chemical in the body that essentially activates it. So no, it's not light. And it's chemical agents that do this. Pridopidine is the most selective one, because many drugs can touch the sigma-1 receptor, but they have broad effects on many other targets. And some targets act to negate the effects of the sigma-1 receptor. So it's a balance. And what's particularly unique here is the selectivity. In other words, you don't want to have a drug that activates target A, which sigma-1, which does good, and target B, which is another target that either antagonizes the effect of sigma-1 or does some separate injurious effect. And we know of a lot of drugs that activate sigma-1, but then activate other things, and they're not as effective. So what's, I'd say, quite and this drug is very selective against sigma-1 in contrast to other targets.
Thank you. So this is targeted to sporadic ALS, but could it benefit someone who has genetic SOD1 ALS? A chart seemed to indicate so. Michael, maybe you answer that one.
Yes, I think that the animal the models that we've looked at are the SOD1 mice, and so this looks like that was where we got most of the data. We also have some data from the C9orf, but not at the animal level, the cellular level, so it's not as robust, I'd say. But I would say certainly the data that we have would predict that SOD1 mutations, patients with these genetic changes would be amenable to this therapy, but not only. We think this would be quite broadly applicable in terms of its effects on many pathways that we've shown.
I'd add that in the platform trial, it's open to anyone with sporadic or familial ALS. But obviously, if someone has a familial form, it'd be really important to discuss other options with your neurologist, because there are some gene therapy trials that are targeting specific forms of familial ALS. Jeremy, I thought maybe you could answer this one. That's kind of related to the SOD1, but is Is the SOD1 mutation what distinguishes genetic ALS from sporadic? If so, do SOD1 mouse studies have bearing on sporadic ALS?
That's a great question, and I mean the simple answer is no. There are actually many different mutations that are now known to cause ALS, and the SOD1 mutation isn't even the most common. The most common mutation is one called C9orf72. That probably accounts for between 50 and 60% of all inherited causes of ALS, where SOD1 accounts for maybe 20%. And there are probably 30 to 40 other much more rare mutations that also cause ALS. Because SOD1 was the first mutation discovered, it was the first one that was actually modified into an animal model. And the model is a good one in the sense that it actually mimics the human disease in lots of ways. The animals grow normally till adulthood and then become weaker and ultimately die, probably a respiratory death. So it's similar in terms of what it looks like. Whether that genetic model actually is a great model for all forms of treatment is a really good question. And the answer to that is probably no. There are things that we know about the SOD1 mutation that are completely different than almost any other kind of ALS. For example, although ALS is associated with abnormal proteins that accumulate in the neurons of all patients, all models with ALS and patients with ALS, for SOD1, the accumulation is SOD1 protein. And for virtually all other ALS, including all the other genetic forms of ALS, it's a different protein called TDP43. And so what's happening now in preclinical studies is often that SOD1 is still a good model to look at whether there's a signal. But more and more frequently, investigators are looking at a whole host of different models because there are animal models of ALS that come from these other mutations. And so I think it's probably true that the broader evidence you can get that your agent is active against a variety of genetic mutations, the stronger your evidence to move into people really is.
I'll add that our therapy selection committee, when we were reviewing the applications to join the platform trial, required evidence in more than one model, just what Jeremy said. It could be the SOD1, but not just the SOD1, and robust target engagement, which you saw Dr. Hayden present. So Michael, this is for you. In Huntington's disease trial patients, have you seen greater slowing if pridopidine is started earlier in the disease process?
Yes, great question. So when you look at the patients with Huntington disease, it's a long disease. It can go up to 20 to 25 years in its full duration. And in the first trial, it wasn't separated to early HD versus late. And what we saw is that the earlier you treated, the greater the effect. When we looked at people with late in other words, quite long, more than 7 years duration, the impact was less. But the problem is the ability to measure using the scale is really particularly useful for early HD. So in early HD, you go down, say, 1 a year. In late HD, you go down between 0.1 and 0.3. So if you want to see, you need many more patients over a longer period of time with late onset. So I would say at the moment, we don't know, but we do know that we've seen the effect very robustly in early HD. And certainly it makes sense if this is not going to regenerate neurons, but rather protect against additional injury. It's better to treat when you don't have very massive neuronal loss. And in late Huntington disease, the brain is quite diffusely involved with neuronal loss. While it starts off selectively, it's broad. And so early would be better. We don't yet know and have sufficient data, and there's no trial that's looked at late onset Huntington disease. And that would need a different trial design for a much longer period of time.
Thank you. These are great questions. We have about 20 of them left. And so I'm going to go through them fast now so we can get some
Okay,
the
I'll answer
answers.
fast too. Yeah.
And then why was a dose of 45 milligrams twice a day chosen?
These are brilliant questions. These are amazing questions. These are all important. So we chose it because we had looked at numerous doses. When you look at lower doses, you get less affinity, less binding, less targeting of sigma-1. And the best targeting, about 95% of targeting, occurred at this dose. When you go to higher doses, you start targeting other receptors, which can minimize the effect. And so we knew from Huntington disease that this was the best dose. And we understand that at higher doses, you're getting targeting of others. And at lower doses, you're not getting full targeting of this receptor. So this was the best dose for targeting. If you want to target sigma-1 and nothing else, this is the right dose.
Thank you. And this is the question that maybe both Michael and then either Bjorn or Jeremy could add to is I noticed that 6 to 8 secondary endpoints in the trial relate to speech or bulbar function. Does this mean that you have clinical indications that this will show more profound improvement in people with bulbar onset or affect?
Well, these questions are very, very discerning and amazing. So I'll just start off. So there is one other drug that's been used in ALS that had significant impact that looks like it's not a very selective sigma-1, as we've explained before. Selectivity is important, but it does target sigma-1 receptor, and that is Nudexta. And Nudexta targets sigma-1, but it targets a lot of other targets as well. But what we did see in some of the early trials, and Merit and others were involved with them, is that you saw some improvement in speech and swallowing as measured by the bulbar scale. So as part of our secondaries, we put that higher because there was some evidence that sigma-1 may be, actually, that may be one of the earliest signs that you might manifest and you might be able to detect. But those trials were short-term. This is long-term. They weren't done. They were different. And so we're looking at that, and we asked ourselves the question as whether sigma-1 stimulation may be manifest very early by changes in speech and swallowing. And that's why it's some of higher up in the secondary outcome measures.
Jeremy or Bjorn, do you want to add to that? You're muted, Jeremy.
As a bit of a caution, I would just say that if you look at the primary outcome measure for this study, it's not solely change in bulbar function. Bulbar function is a huge contributor to disability in ALS, and I think in many ways it has not been well looked at in the past, and so I think it's very appropriate that we look at it in this context. But I mean, the animal data, a lot of the preclinical data suggests that if pridopidine is going to be effective for ALS, it won't just impact bulbar function. I mean, that's not the outcome we're looking for.
Yeah.
Bjorn, is there a test to confirm what is the neuron status, weak, dying, etc., or the normal amount of neurons someone should have?
Not really, no. I mean, we do, after death, when we perform autopsies, we can look in detail at the state of the neurons. I would say that otherwise we can measure if a neuron functions. We have electrophysiological measures of that. But if it is sort of stunned or not doing well, or if it's truly lost, I think it is difficult for us to determine. There may be markers such as neurofilament that would probably indicate that. I guess I would say the answer is no. We can't really truly tell with a good degree of precision if something is not working or if it is actually dead.
A good,
I guess just because I've spent almost all my life partially working on this, I have to pipe in and say there is an electrical measure called motor unit number estimation, which actually gives you an estimate of both the number of surviving motor neurons going to a muscle and how many muscle fibers that neuron is able to connect to. And we have a good idea of what normal is for both of those numbers. So in some sense, we can assess whether the motor neuron complement is normal and whether it's behaving somewhat normally as well.
But you can't tell if they're recoverable when they're lost.
Definitely, that's true.
So, Michael, given the extensive safety data available in HD, is the FDA requiring the same amount from ALS, or will they allow reduced safety data collection required for submission?
What we're hearing right now is that we have, certainly for the HD trial, we have sufficient safety data for a new drug application now. I think the safety data for, even though it's not just for Huntington's, it's been used also in Parkinson's disease and others with no increase in safety. So we do not expect there to be any difference in safety for ALS. But the safety data from this trial will be very important to be able to further support its safety, not only in Huntington's, but in ALS. I would say certainly we will need the safety data over this period, and the open label extension will also help provide additional safety data that will support what would be hopefully a successful outcome.
That's a question. Is there any specific stage of the disease in order to take the medication? For example, if someone has a tracheostomy or is completely paralyzed, is there a possibility to reverse in this stage?
Well, the answer is we don't know. Based on everything we've said, earlier is probably better than later, but we don't, and if neurons are injured, there's still an opportunity to halt the further injury. I don't think you're going to start seeing people have complete recovery of function, but perhaps there'll be some delay in progression. And just to follow on what Jeremy says, of course, the functional progression is what's the primary outcome here. And hopefully these patients will show no further progression, but I'm not expecting them to suddenly develop mobility with this, but hopefully slower or less progression.
So if a trial participant showed positive results, how long would you continue to provide access to the drug?
Well, I think we would then, in the presence of positive results, we would have a very extensive open label that would go on for an extended period of time. We haven't yet discussed that, but obviously there would be a desire to keep patients on this as long as possible, certainly until the drug is approved. And so this would be extensive open label, but we don't yet have a decision on that particular issue. I may just add, of course, there are strict criteria for inclusion and exclusion in this study. And if somebody does not meet the inclusion criteria for whatever reason, we are in discussions with Merit and other colleagues about providing expanded access to this drug as part of a separate protocol. And we will be doing that, but the details of which are still being worked out. But we also want to be able to provide this for patients who, for whatever reason, don't meet the inclusion criteria in this trial.
There's a question that I think is related to your comment about the time for maybe FDA approval for Huntington's. I think it's probably worth answering because we don't want people left thinking that this is going to be 5 years from now. The comment is that the average lifespan might be 2 to 5 years for people with ALS. So 5 years is like a lifetime for people. Anything that can be done to speed up the process. And I'll just say from the HEALEY platform part of it, we've had many meetings with the FDA before we finish the design and launch. And we have the commitment from the FDA to meet with us as many times as we need. And we did talk about the impact of a single study. And there is a process if a single study is robustly positive for faster approval. So they have been helpful with us on that. So first we have to finish enrollment and get the results. But I just wanted to put that out there for people to know.
Yeah, and I will just add to that, if there was a successful, a positive, compellingly positive result, we would expect accelerated approval in a much shorter period than we're going to see for the Huntington trial, which is going much longer. So this would be a much shorter approval, and in this unmet need, this would go to high priority for the process of regulatory approval.
There's a couple of questions asking whether there's a subset of people with ALS where, based on the mechanism of action, you might expect to see efficacy.
So, I mean, we don't really know that at this point. You know, we've shown from the New Dexter data, we wondered about whether patients with bulbar abnormalities would show this, but we would expect this to have generalized effect in terms of function. So we don't really have a prescribed view on which patients would show benefit. The mechanism involved should show overall generalized benefit to essentially all patients, irrespective of the cause, because these mechanisms are involved. So of course, we're going to look at that as part of the statistical analysis, and this is all being pre-specified. But at the moment, we're hopeful that, you know, this will show an improvement or a slowing of progression in patients, irrespective of the cause and the symptom.
Thank you. There's two platform-related questions I might answer, and there was a third one that I answered by writing, but there were questions about inclusion-exclusion criteria, and I just want to share that those are posted on the HEALEY ALS Platform Trial website and clinicaltrial.gov, and we show them on our other weekly calls, so we didn't do it today. In the platform trial, will combination therapies be tested? Example, A plus C. And that's a great question. That is the beauty of these platform trials, that if you get a positive trial, you could do those combinations. And that's certainly in our mind, in the group that Jeremy and Bjorn are on, thinking about design. But the first thing you have to show is that that drug is positive. And the other one is about our plans, patient enrollment, pace, and schedule. And so enrollment was actually really fast. It's still going very well. It has slowed down a little bit, and I think that is driven by the global level, the pandemic, and hopefully it'll pick back up. But the hope is that enrollment for these first regimens might be done in the spring. And then again, we wouldn't have results until the last person finishes 6 months. I might want to just give any closing remarks from the co-leads for Pridopidine, and then Michael after that.
Well, I just would say that these are great questions. Thank you for your interest. I think this is a really promising drug. I think it's really important that we're testing it, and I'm looking forward to seeing the results like everybody else.
And I would echo all that. And I think that, again, this platform methodology is very well suited for quickly moving through drugs, so we get a solid hit with something that really makes that difference that you guys were asking about. This is how we can get there the fastest.
And I'll just say that, you know, it's a privilege. I've been amazed to see 143 people participating in this discussion today. The interest is overwhelming. I appreciate the trust and the interest. And I would say that I've spoken about Merit and Sabrina, but it's also a privilege to work with Bjorn and Jeremy, who are outstanding PIs for this particular study. So we look forward to talking to you again and working with you to assess the drug in this indication.
Thank you all for joining. We'll record it and post it and answer the questions by email if we didn't get to your question. Thank you very much. Have a good evening.
Thank you.